Sweet.
Okay.
All right. Well, welcome to another version of Systems, Neuroscience, and Complexity. It's a real pleasure today to have Taylor Bolt and co joining. We have a really cool little consortium of scientists joining on the call today. So I reached out to Taylor as part of a discussion I was having with Shella and her group about spatiotemporal dynamics and neuromodulatory systems and all other kinds of fun things that we think about. And just around the time that we reached out to chat, this beautiful paper dropped from your group, and it's really close to a lot of the things we think about. So we're really eager to kind of hear about, the process that went into coming up with that idea and kind of the insights that you've developed from it. We're big fans of thinking about, basis sets and what the best basis set would be so that we can start to really kind of compare and contrast across groups. And all these little idiosyncratic things we find, it's often very hard to sort of put them onto a sort of notice board together and figure out how they link together. So having these kind of approaches where you can find a really simple way into a complex set of results and then start to pin them together is really attractive to us as a kind of interdisciplinary group here in Sydney. So really excited to hear what you've been up to, Taylor. And I should just mention to everyone, Taylor has to go in 40 odd minutes. And so at that point, Shella might take over. We'll sort of see how the day takes us. Cool. All right. Thanks, Taylor.
Of course. All right. Can you all see the screen?
Yes.
Yeah. Oh, okay. Yeah. Awesome. Okay. Good deal. All right. Well, thanks, Mac, for the invitation, and I appreciate the interest in the paper. And of course, we got our co-authors here, Lucina, Katie, and Shella, so they'll help me out and hopefully have a great discussion. And actually, believe it or not, we actually have some new results we want to bounce off y'all, if that's okay. So we'll talk a little bit about that later. But so the title is a bit of a mouthful. I won't go through it, but it's fairly descriptive of what we've been trying to do, which is develop something like a taxonomy of functional connectivity patterns. That was the real sort of generating idea of this. If you hold on, let me see. Can I get there? We go. So the real impetus was, really the complexity of the functional connectivity landscape. You can pick up any paper and just see a number of different ways of representing functional connectivity. And so if I've got a sampling here of some of these, and I can't get into all of them, but into the details, but I'll just give you the titles. We've got functional connectivity gradients. We've got metastates, which are derived from hidden Markov models, temporal functional modes, quasiperiodic patterns, the task positive, task negative, that's been around for a while, lag projections, coactivation patterns, functional networks, which is the literature in and of itself, global signal, and you can kind of see where I'm going. It's a complicated literature, and really you can have kind of two attitudes to this, and I'm going to borrow, Lucina, if it's okay, kind of two terms that you used. You can either look at this and be a splitter, or you can be a lumper. So a splitter looks at, is looking for those unique insights that might come from a particular representation, while a lumper is looking more to like put all this together, put it into like a tidy boxes, and kind of see see it all together as, in a small categorization. And I'm a lumper. I don't know if it's like a scientific impulse or it's more of an OCD thing. I don't know, but that's I like to see organization. And so the idea is we would develop something like a taxonomy, not of, and this is important to make this point to make, not of the mathematics behind all this, because I think, that's complex. That's been covered in plenty of other literature, but more about the patterns themselves. So can we get a grouping of these patterns that are, into a small categorization? That's the idea. So less of us trying to find any fundamental relationship between like the algorithms behind this, but more of the representations. So With that said, we do make a kind of a fundamental distinction between those two, between these functional connectivity representations from the outset, particularly in the information that they encode. So really the information in terms of the synchrony that's being measured by the functional connectivity representation. So we distinguish zero lag synchrony, which is probably the most common way we study functional connectivity, which is just simultaneous statistical dependence. Something like a Pearson correlation coefficient, no lag, zero lag. And of course, examples of that are here, functional connectivity gradients, networks, so forth. And the sort of patterns you're going to be picking up with this measure are fixed spatial patterns that fluctuate up and down over time. So if you kind of think about that, we kind of think about that in terms of a standing wave, which I've got here. And I'll be honest, I spent an ungodly amount of time trying to get this animation to work. So I hope y'all enjoy it. But what I've got here is a standing wave, a one-dimensional sinusoidal standing wave plotted over a line. The positions of the line are along the x-axis. And then over time, you're seeing the position fluctuate at each point. So the position on the y-axis is the signal amplitude. So if you look at this, and the point is, all of the points on this line are in phase or anti-phase synchrony, okay? So if we were to, so it could be captured by zero lag synchrony. So if I grab this red point and this blue point and I plot them over time, you'll see that, difference in amplitude, but total in phase synchrony. If I would have grabbed something, say at point at 15, you'd see complete anti-phase synchrony. A generalization of zero lag synchrony is something like time lag synchrony. So a statistical dependence between two time series at a lag of one or the other. And of course, that could be zero lag, in which case you would have zero lag synchrony. Examples of those that can capture that information are quasiperiodic patterns or lag projections. So in terms of the patterns you're going to be capturing with this is, well, not just fixed spatial patterns. You could if you wanted to, but you could also capture patterns that evolve over time. So something like, it could be a propagating pattern or something. So something like, a traveling wave. And this is the same animation. And now it's just the same one-dimensional sinusoidal oscillation, but now it's a traveling wave. And if we grab those same points, you're seeing same amplitude, but now these are phase shifted in time. So you're going to need something like, in order to see that relationship, you're going to need to do like cross correlation or coherence or something. And those are the preliminaries. So with those preliminaries out of the way, we can tell you kind of our primary findings, and we'll get into them. In terms of the primary findings, many functional connectivity representations are just describing different facets of the same underlying spatiotemporal patterns. And I don't want to say all, because we didn't study all of them. We just studied as much as we could. They did seem to resemble these spatiotemporal patterns. And two, a small set of these spatiotemporal patterns can explain a large number of previous findings. So a large number of previous functional connectivity representations. So that's kind of the overall findings. And in order to get to the first finding, capturing these spatiotemporal patterns, we wanted to develop a way of capturing both zero lag and time lag synchrony in a single representation. And what we did was we actually borrowed a technique from the geophysical sciences, what we're calling complex PCA. It has a bunch of different names. And, I'd gladly go into kind of the literature review behind this, because there's a lot of cool stuff this has been developed for, and it has different names, but I'll unless you guys want me to go into that, I'll just avoid that for now. But complex PCA is really actually not all that different from PCA. You're just adding a little bit of extra information to the signals before you do PCA. And so I put it out in these 4 steps. So the first step is what you would call the creation of an analytic signal, which is just the original signal plus it's what's called its Hilbert transform. Those together in an imaginary number, you combine those two into an imaginary number. You know how you back in grade school is A plus BI, how that's typically represented. That's what you're going to do first, and that's a very common signal processing technique. And one way to show that is with a simple sinusoid. So I've got like a cosine fluctuating over time at a single frequency, and the Hilbert transform, at least in this case, has a very obvious interpretation. It's just that signal shifted by a 90-degree cycle. Okay. For cosine, we know what the signal is. That's actually a sine wave. And if you put that together, you get into an imaginary number, you get imaginary number, right? So the real part is the original time course, and the imaginary part is that time course shifted by 90 degrees. Okay. Now I'm going to admit that sounds really odd. I'm not a signal processor or a mathematician, but what it facilitates is a mathematical representation that's very useful. And that is Euler's formula, right? Have y'all seen that before? It's funny. I don't know where I read this, but I guess some magazine, and they asked mathematicians what's the most beautiful formula they've ever seen. And by and large, it was Euler's formula here, which is the relationship between a complex signal here and a complex exponential. But what that complex exponential is giving us is really this. It's giving us magnitude over time, right? This R. In this case, this magnitude isn't fluctuating, but in a complex signal, you'd have that. And an angle. So where are you in the cycle of that oscillation, right? And this sort of kind of explains the need for band passing your filter. An angle doesn't really, like where are you in your cycle doesn't really make sense when you have a frequency or a signal composed of like a wide range of frequencies, right? Now that's okay because you generally do in resting state band pass or filter into a slightly narrow range. But all of that said, you're creating a complex, an analytic signal. And what you're going to do, and you're doing that for all your brain signals, and then you're just going to create a correlation matrix, just like you would, any other functional connectivity matrix, symmetric brain region by brain regions. But in this case, it's when you take two complex vectors and create and do a correlation coefficient between them, you're going to get back a complex number. Okay. So for example, if we have brain region X and Y, you have, the simple representation A plus BI. This is what you'll get back. You'll get back a complex number. This number isn't that interesting in and of itself. If you want to know, the real part is the correlation between the original time courses, and then the imaginary part, the B, is the correlation between the original signal X and the signal Y phase shifted by about 90 degrees. Okay. Now, I mean, that's information, but it's not all that interesting for what we're looking at. But what you can do is essentially what I did up here is this a polar representation of that signal. And then again, by Euler's identity, you can get a magnitude and a phase. And what this is it's really cool when you do this complex correlation. You can calculate the phase difference between those two regions. And you also get a magnitude component, which is the absolute value of the correlation at that phase difference. Okay. So those two steps are really the only different steps you would take for PCA. I mean, at that point, you would just apply regular PCA to this complex correlation matrix. And it's the same interpretation as PCA and regular old, non-complex numbers. So the first dimension you'll pull out is the axis of that explains the most variance and so forth. And the second, of course, they're all orthogonal to each other, spatially orthogonal if you're doing a spatial PCA. But what's cool about these principal components that you get back is they're filled with little just nuggets of information about about spatiotemporal information. So if you look at the output of a PCA, you get like spatial weights, right? At each voxel. And what that is just the correlation between the voxel time course and the principal component time course. You can read those and see what the pattern's all about. Now, because this is a complex signal, so you're getting a complex correlation back between each voxel and the principal component time course. So we can take the magnitude of the principal component, we get the magnitude. We can also get a phase delay map. So we take the phase delay between each signal and the principal component time course, and you can get a temporal evolution of that signal over time. And then, of course, you can take the real part, which would be the correlation between the original time courses and the principal component time courses. Then you get the imaginary part, which is, again, same interpretation here. It's the correlation between the original time courses and the principal component time course shifted 90 degrees to forward in time. And then, of course, you get, complex time courses as well, which you can take the magnitude of as well as the phase. So I'm doing a lot of talking. Any questions about complex PGA and how that all kind of operates? And hope this was clear as mud. I don't know.
Can I just ask a quick summary question? So the Hilbert transform is basically transforming the signal to itself and a second dimension, which is itself with a pi on 2 phase lag. Is that basically it?
Yeah. Well, okay. In a simple sinusoid, that's it, right? But if you have a signal with a bunch of frequencies in it, it's going to be shifting that frequency across all the frequencies.
Each of them, right.
Yeah.
And so, in some sense, the way I see what you're doing then is that you're effectively creating an embedding for each of those frequencies, where having itself with no lag and having itself with pi on 2 allows you how those two compared to any other signal gives you a representation across all the possible lags there could be between those two signals, doesn't it?
Yeah, well, Joseph, I'll tell you, that's the best intuition. I haven't thought of it that way, but that's a really good way to communicate that. So I appreciate that. I'll have to start using that, because it's kind of hard to build that intuition from here to pulling out spatiotemporal patterns. So I appreciate that.
Okay, I'll shut up. I've got more questions, but I'll shut up because I know you got limited time.
Sure. Well, okay. So we took complex PCA, and we applied that to resting state data from the Human Connectome project, and we pulled out 3 patterns. And I'll tell you why we pulled out 3 in a little bit, but we'll just look at the patterns. So what I'm showing here is the first complex principal component, so the component that explains the most variance, and we just call that pattern 1. You can look at the phase delay map, which is the phase delay between each signal time course and the principal component time course. And if you look at that, and we have it mapped to color here, you can kind of see the temporal evolution over time. And I have it in a circular color map because phase is actually, it's a circular statistic, right? So you go from 0 to 2 pi, you're back at the same number again. So if you want to read the temporal evolution, you would go counterclockwise. So you would go, okay, here's where it starts. Sensory motor cortex, superior temporal cortex. You see it propagating outward. You're in the green. Move up to the blue, and then it finishes there in the pink. Now, I'm going to be honest, these are so hard to for me, it's so hard to map color to time. So what we have to the right is actually actually sampled time points of this pattern. And you can see the dynamics of what I'm describing in time. And that's pattern 1. So it's fairly global. And just to anticipate, that is statistically, the time course of this is statistically identical to the global signal. So if you were to take the average gray matter time course, this is what you would, and you maybe averaged around peaks of that time course, you would see something like this. Second pattern is a fairly anti-correlated spatiotemporal pattern. And if you look at this, it's essentially mapping on, there's the default mode, and then there's the task positive. So that is essentially mapping to the task positive, task negative pattern that's been observed in previous literature. Now, pattern 3, we didn't really map it to anything. We couldn't really find a clear example in the literature of someone, saying this is some sort of pattern, and we were able to map it back to. But it seems to have, it has anterior cingulate there, has anterior insula and so forth. So, who knows what that is, but the way we decided to extract 3 patterns is based on a simple test, which is just a scree plot test. I think it's called Cattell's scree plot test. And the way you do that is you just plot the eigenvalues, which are essentially the amount of explained variance of each component. And you just plot that over time. And you're looking for a point with diminishing returns. So there is a strong elbow at 1, or, right after 1, and a strong elbow right after 3. Now, 1 would just be not that interesting. We wanted to extract 3, so that's the reason we chose 3. But I think the point here, and I think we tried to make it in the manuscript, was we're not saying that there are just 3 spatiotemporal patterns in resting state. We're just saying that you can do a lot with just these 3 patterns. So you can explain a lot in terms of previous literature, and I'll get into what I'm talking about in a second. But related to that, so we've been talking, there's a lot of time lag here, but there's also a lot of fixed spatial patterns here, especially take a look at that. I mean, there's some propagation that you're seeing, but it's really oscillating between 2 fixed patterns. Same with a lot of these, maybe a little more propagation here. So it really is, these are really composed of predominantly standing waves. So we actually developed the metric and measured that. And these are predominantly standing wave patterns. And that should tell you something, because if it's predominantly standing waves, then zero lag methods that don't even account for time lag should be picking up the same signal. And in fact, this is complex PCA. If I just run a regular PCA, you should be able to see the same patterns. And that's exactly what we find. So if you just do regular PCA, you're going to pull out the fixed spatial patterns of those. And, you can see that visually, but, we also look at the spatial correlations between those maps, the time courses, and they're all very similar. Now, in terms of a taxonomy, if this is, what we want to go with, the question is, well, how well does it explain previous literature, so previous spatial patterns? And what we did was we just did a survey of a bunch of different zero lag functional connectivity methods. So coactivation patterns, hidden Markov models, ICA, spatial ICA, temporal ICA, a couple other things. And we just said, okay, let's grab those spatial patterns. And we did low dimensional solutions, so about 3 for each solution. And then we correlated those spatially. So just took the spatial correlation between them and the principal components. So there's the fixed patterns. And what you see is, by and large, they map pretty closely to the principal components. You're getting very similar findings. And essentially you're just getting consistency across these. Now, there are differences there, which I think is something we didn't really talk about much in the paper, is that, yes, there are differences. They are very consistent. They're all kind of giving similar patterns, but at the end of the day, which one of these, because they're all different slightly, which one of these is an optimal representation? Is there a true or false here? Is there one that's closer to the real versus not? I'll be honest, I don't really know the answer to that. We can say that they're pretty closely to the principal component solution, but which of these should you take and start probing downstream for these patterns? I'm not totally sure. I'm curious if anybody has any thoughts on that.
Can I ask a clarifying question that may help?
Yeah.
Did you bandpass filter this data in a particular frequency band? And if so, did you mess with that a little bit to figure out whether or not letting in different timescales might expose more or less non-stationarity?
Yeah, we did not. So we filtered to the straightforward range of 0.01 to 0.1. But you're right. I think there could be some potential uniqueness to be found, or divergence in these representations if you look at another frequency range or a broader frequency range.
Yeah, cool. The other thing I found myself wondering was what it would look like if you had someone, you know, cough at some point in the scan, which as anyone who's played with HCP data knows happens probably more than you would have liked. Did you have a look at, you know, closely, I'm sure you did, at things like, you know, big abrupt movements and see whether or not that had a big effect on the kind of loading in this space?
We didn't, in this study, we did not take a look at any like mapping to any scanner behavior. But actually, if we get to those new results, we actually have tried to map these to both electrophysiological signals and physio signals.
Oh,
So,
awesome. Yeah, cool.
Yeah.
Right. I won't delay you too much. I'm definitely on the camp of not kind of aggressively removing those things from the data blindly,
Right.
because I think there's such a strong integration of physiological measures with a bunch of neuromodulatory systems that end up having a massive impact on, you know, cognitive dynamics. And so I don't want to kind of like throw out the baby with the bathwater, but you always worry that people will come back and show some video of someone, you know, shifting their head, and then all of a sudden the PCAs go nuts and you're like, oh, wow, it looks just like when they were thinking. And it's always that concern in the back of your head.
Actually, Taylor, you might even consider skipping to those new results just because you don't have much time. And also,
please do. Yeah,
everyone can read the paper. So, I mean, I'm actually curious what this group thinks of your new physio results, just because, you know, we've had less time to think about that, really.
Yeah. Okay. Yeah, I'll go straight to there. So where it all started was, you know, we had a discussion potential where all these come from. And we offered some potential explanations, some more physiological than others. Things like potentially systematic vasculature. So if you've heard of systemic low frequency oscillations from Blaise to Frederick, is he at Harvard Hospital?
Yeah, McLean.
Okay. Yeah. So they've identified this cerebral blood flow pattern that seems to co-vary with a peripheral blood flow, which, you know, they get from a signal of apneas, I think, on the finger. And this is a large scale pattern that co-varies with peripheral blood flow, which is the idea. If it's co-varying with peripheral blood flow, then it's got to be systemic to the vasculature. There's also low frequency respiratory changes. As we know, those are causing widespread changes in bulge signal. Of course, there's the ascending arousal system, which, you know, would presumably have fairly global effects. And then recently, there's been this talk of these global bold and CSF oscillations. Have y'all seen the Fultz et al. 2019 science paper where they essentially showed during sleep there's these high magnitude fluctuations of CSF?
Is that from Laura's group?
Katie, who's this group from?
That's Laura.
Yeah, Laura Lewis and MGH.
Yeah. We know that. I just didn't know that one as the first author. Yeah, we know that stuff. We also saw a really cool talk by Ryan Rort, who was working with Mark Reichel for a while. He had a Science Advances paper where they took physiological data and then did like a phase analysis and then yoked bolds to the kind of peak and fluctuations in like respiratory rate and heart rate variability and things like that. That was really cool. It's along these lines.
Yeah, we'll have to take a look. Yeah, I think I remember reading that, but I'll take another.
You gave a talk on our YouTube channel if you want to put it on the background. It was really nice.
Sure thing. So I think that one thing that I think about when proposing all these changes, and Katie, you'll probably nod at this, is like, really, these are all, you know, integrated physiologically. So I put this, you know, little web together. You know, all the different signals that are correlated with each other, that are correlated with really what's been systematically studied as pattern one, which was what we found to be the global signal. So there's just all of these signals that are fluctuating together with the global signal. And so it's really hard to, I guess, untangle that web, just isolating one. And so I think the best way to get at this is just to record as many of these as you can and see how it varies in different states. And thankfully, Katie has supplied with a supplied us with a multi-echo fMRI data set with simultaneous EEG, and we have looked at that, and we're also using HCP and another multi-echo data set with Physio for replication. And so kind of generally what we've found, and there's a lot here, and I'll kind of walk y'all through it, is that, I mean, these are, the pattern one is correlated with a bunch of different physiological signals. First, if we look at kind of the CSF, so this is the volumetric representation of pattern one. So what you were seeing on the surface earlier, this is in volumetric space, and so we're picking up ventricles and so forth. But what's important, I think, it's a reason, you know, kind of to take the cortical maps with a grain of salt, is we saw a very global signal in the cortical, but if you allow in the ventricles, you see this anti-correlation between the global, the gray matter, and signal coming from the ventricles. And we also see that, of course, with the cross correlations. And so what this cross correlation is the thick line here is the average, or is the median cross correlation, with Katie's data set in blue and HCP in red. And then all the individual lines are the individual subjects. So you're just getting an idea of the variability. So we're seeing that sort of anti-correlated fluctuation between CSF and this pattern one. Another thing to look at is the time delay. So the phase delay of that pattern one when we look at it in volumetric space. And it's fairly consistent with what we were seeing, but again, I think it's important to look at it in volumetric space because you get a little more insights. Again, it's kind of in the sensory, motor, visual and stuff where things start. And I'm following this counterclockwise. So the dark orange, and then follow to the white. You see precuneus, anterior cingulate. But you also see, which is fairly, which was, you know, very observable and Tong or Blaster-Frederick's study of the systemic low frequency oscillations is this is getting pushed back to the draining veins here. So we have a brain mass that's dilated, so we'll pick up some of that. And then, of course, you see the CSF ending is the last thing to end. Now, another reason we wanted to look at this volumetric space is we wanted to see if this time lag structure mapped to the systemic low frequency oscillation, which is a pattern of time lags that correlates with a signal in the periphery, which is just a peripheral blood volume signal. Now, in principle, they did an FNIRS, but you can get that signal as long as there's not a high pass filter on the hardware. You can get a low frequency signal from that PPG on your finger, the same way that Blaster-Frederick got it from the FNIRS probe. And so, so when we take that time lag and pull back, you know, what they were seeing, the systemic low frequency oscillation, which is, you know, the time lag that co-varies with the peripheral blood volume signature, you see that, you know, despite the change in scale, so that the colors are slightly different, they're mapping precisely onto pattern one. So if you take the maximum cross correlation between a peripheral blood volume signal, low frequency, and across voxels, compare that to the time lag structure of pattern one, you're seeing very similar mappings. And you can see that in the cross correlations between these two. They're very strong. So another thing that we wanted to look at, and so I'll just let you all look at some of these cross correlations. They're fairly strong across a lot of the variables that we measured. So we measured respiratory volume, heart rate, pulse amplitude from that PPG, and then of course we had simultaneous EEG, alpha. We pulled alpha, we pulled infraslow, which I now am a little concerned that it may not. I don't think it was necessarily set up to pull infraslow EEG. It might have not been optimized there. So take that with a grain of salt. But what we're showing is across data sets is that pattern one, its time course, is associated with a lot of different physio signals. And what's interesting, it's important for later, is that they tend to have bimodal peaks about 10 seconds separated in time. So if you look at this, take a look at the heart rate, take a look at respiratory volume and so forth. They tend to have those bimodal peaks at 10 seconds, which I'll come back to, and I think it's relevant. Now, another thing we wanted to do was, if there is, these are just individual correlations between PC, between pattern one time courses and each of these individual time courses. So, that's not really showing that there's this joint co-fluctuation, that they're all going up and down together. So what we did was just a joint correlation analysis to demonstrate that, which is essentially canonical correlation analysis. An extension to more than two sets of variables is what's called a multi-set CCA. So we put in all the physio signals and all their different and their lags as separate sets of variables, and then we will find a latent variable that is maximally correlated across all of the signals. And what you see is, if you pull the first component from that, you extract a component where there's just a lot of correlation between all these signals. So I guess what we're trying to say here is that all of this stuff seems to be going up and down together. So it can be hard, I guess, to potentially find out what's the origin, right? But we do have, of these signals, we do have one of these where we can easily manipulate. And what that is respiratory volume, right? So we can manipulate respiratory volume through some sort of, task or something, and see what happens to the rest of, the bold and the physio. So what happens when we do that? So Katie provided us with a kind of a sparse event sampling inspiration task. So, you're sitting in a scanner, you are, sitting there for a while, and then you get this auditory cue, and then you're directed to take a deep breath. And there's about 9 events to those. And so what we can do is, we have simultaneous EEG and physio with that data. We can just take a trial average to see how everything looks. And of course, when you do this, take this deep breath, you see this time course for the first principal component. Now, remember, the first principal component, or the pattern one, I'm using those interchangeably, is fairly global. So one thing to keep in mind is the respiration response function was global too. It was just a global average. And so what we're seeing is the first principal component pretty much maps precisely to the respiration response function from that Gary Glover and Katie have looked at. And again, sorry, these are, the x-axis here is TRs. So you got to multiply that by 2. So the TR was 2. And so you'll see that those map to each other. But what's interesting is you see there's that early increase and then a late decrease. So you can kind of distinguish those into 2 phases. A global bold increase, so the pattern one increases. We see an increase in alpha arousal, or alpha amplitude. We see an increase in heart rate, and then a decrease in low frequency peripheral blood volume. Now, I'm not a physiologist, but in terms of like potential explanations of what that peak is, I'm seeing a lot of potential signatures and maybe sympathetic activity. So sympathetic activity maybe causes a jump in arousal, so you'll see an alpha EEG jump, and then sympathetic nervous system activity. High activity leads to peripheral vasoconstriction or vasoconstriction. So that would be consistent with the signal decreasing for the low frequency PPG component. And then for the late phase, it's fairly consistent with the typical story of respiratory fluctuations, which is when you take a deep breath or hold your breath, you're changing the CO2 concentration in the vasculature, which CO2 is a vasodilator. So If you take a deep breath, you're going to dilute some of that CO2 and cause a decrease in global bold. And if you take a breath hold, you should see an increase. And that's kind of what we see. And then for the physio, we see an increase in CSF at that time point, an increase in heart rate, an increase in peripheral blood volume, and PPG amplitude. So, I mean, I guess what I'm getting at here is that I think there's two phases here to this, response to respiration that's mapping onto different physiological systems. That's what I'm trying to figure out. Any questions on that?
I found myself curious what the second and third patterns look like with respect to the physiology. Do you see anything?
Yeah. So we're in the midst of trying to bring that all together. I will say, at the very I've got to run in 2 minutes, but I can, Max, share this the end of the deck with this with you. We are seeing a mapping between both pattern 1 and pattern 3, that they seem to be just time the time lags of each other. So you see a pattern 1 increase and then a pattern 3 right around respiration. So they're kind of time lag to each other. In terms of pattern 2, the anti-correlation, we haven't really figured that out. So we're still trying to figure it out.
Yeah, cool.
All right. So
yeah, I was going to ask you, I'm glad you kind of got there with the sympathetic stuff. It'd be fun to sort of have a think about how you could augment or diminish that system in a slightly more complicated sense with, different medications. And there are a couple groups, in fact, Shella will probably think about this with her stuff already, but looking at that LC stim work that you've got, Shella, would be really cool, right? With the opto. I don't know how this will be affected by anesthesia. Obviously, the mice that have the optogenetics in the simulation are lightly anesthetized, and so it raises a whole other set of questions. But causally impacting the sympathetic nervous system would be really cool to see what happens.
Right. Shella, yeah, well, we definitely have to. I know Shella has a lot of potential data sets to look at that, and so I think that'll definitely be a next step for us. And awesome. Then the last thing, and then I'll jump y'all. So there's kind of two phases here in the response to respiration in the physio. What that suggests to me is you should see a difference in spatial pattern of the bold at this early phase versus this late phase. And that's precisely what we see. We extracted that in two different ways what I'll say is, if we look at, ignore this at the top, I'm just going to show you this down here at the bottom. If we looked at the trial average of that in terms of just the bold signals, you see this sort of anti-correlated pattern here, followed by the global decreases, what you'd expect from arterial CO2. And the general story here is there is a different spatial pattern of bold right in the early phase versus the late phase, where you start to see this global increase. And so we're still trying to map that out, but that's kind of the lesson there. And at this point, I will have to hand over the reins.
I can